The failure rate of immune prediction. A single drop of blood contains over 10,000 unique T-cell receptors, but predicting which one will latch onto a specific cancer mutation has a failure rate of 99%. PanPep AI is moving that needle by treating the immune system as a meta-learning problem rather than a simple classification task. By training on how the body learns to recognize new threats, this model identifies effective T-cell targets with over 90% precision across rare genetic profiles. This shift from massive data crunching to structural reasoning is what makes the recent Nature Machine Intelligence benchmarks actually matter for vaccine design. The core of this capability lies in how the system handles the sparsity of human genetic data. Solving the HLA sparsity problem. The primary barrier in immunology is the human leukocyte antigen, or HLA. There are thousands of HLA variants, and for the vast majority, we have zero experimental data to train on. Standard deep learning models fail here because they require thousands of examples per variant to learn the binding rules. PanPep sidesteps this by using a meta-learning framework. Instead of learning specific peptide sequences for one person, the model learns the underlying physics of how any HLA molecule interacts with any peptide. It essentially learns how to learn a new HLA profile from just a handful of data points. This meta-knowledge allows the system to generalize to rare populations that were previously invisible to computational biology, and the underlying mechanics are surprisingly efficient. Meta-learning versus standard fine-tuning. While many assume this is just another fine-tuned transformer, the architecture uses a support set and a query set to optimize the initial model weights for fast adaptation. This is an optimization of the gradient descent process itself. When the model encounters a new virus or a patient-specific tumor neoantigen, it doesn't need a full retraining cycle. It performs a few-shot update that adjusts its internal representations in seconds. While some critics argue that the model still struggles with highly non-linear binding motifs, the zero-shot performance on unseen HLA types is currently beating standard neural networks by over 30% in F1 scores. This speed and accuracy are now being tested against real-world biological noise, the benchmark reality check. We have to look at the edge cases, because that is where personalized medicine lives. In the validation trials concluded in April 2026, PanPep was tested against the IEDB benchmark, specifically focusing on the hardest-to-predict Class II molecules. While the 90% accuracy holds for common alleles, performance still dips to 75% when dealing with highly mutated viral sequences. This suggests that while the neural digital twin is excellent at predicting structural fit, it still lacks a complete understanding of the kinetic stability of these bonds. However, comparing this to the previous industry baseline of 40%, the delta is significant enough to reduce the time for personalized cancer vaccine formulation from months to weeks. This technical efficiency leads to a broader shift in how we approach vaccine engineering. The shift to deterministic immunology. The success of PanPep confirms that the future of biocomputation isn't in larger models, but in more efficient meta-architectures that mirror biological flexibility. By solving the few-shot learning problem for T-cell interactions and bypassing the HLA sparsity issue, we are effectively moving from a trial-and-error approach in oncology to a deterministic engineering discipline. For developers and bioengineers, this means the bottleneck is no longer data acquisition, but the integration of these high-fidelity predictions into automated synthesis pipelines. If these 90% accuracy rates hold in the upcoming clinical phases, the standard of care for late-stage melanoma will shift toward purely computational antigen selection by early 2027. We are transitioning from predicting if a vaccine will work to calculating exactly why it will. Watch the results of the upcoming Merck-Moderna collaborative trial in August 2026 for the first real-world validation of these meta-learned targets. Subscribe for more